Thanks for everyone who is joining. We will wait for another couple of minutes before we begin the session. Good afternoon, everyone, and good morning to my colleagues on the West Coast. My name is Rosita Wooster. I'm Dean of the Graduate School at Portland State University, and it is my pleasure to welcome you to our final 3MT competition today. I would like to acknowledge and thank ProQuest for their support for this competition. And without further ado, we can begin the competition. Next slide, please. Thank you. As many of you know, the 3-minute thesis is a competition where students have just 3 minutes and only one slide with no animations, and it is spoken. The scoring criteria are available on the WAGS 3MT website, and it is my pleasure today to introduce to you our judges. Our first judge is Amy Scott. She is Associate Vice President for Government Relations and Public Policy with the Council of Graduate Schools. Next, we have Will Lamb, who is Dean of Graduate and Adult Learner Recruitment with EAB. And finally, we have Gilia Smith, Director of Academic Relations, Australia, New Zealand, and North America with ProQuest. So the final thing to note here is today our technical support are Pei Zhang and Mary Braden. By the rules of the competition, we will not have questions from the audience, and so you won't see live chat. And of course, please mute yourselves if you already aren't. So our first competitor, next slide, please. Thank you. Our first competitor today is Kishore Chittimali. He is from Pharmaceutical Sciences Graduate Program in North Dakota State University. His advisor is Dr. Yagna Yarajapu.
Yeah.
And the presentation title is Restructuring Gut Microbiome for Healthy Aging. Kishore, are you ready?
Yes, I am.
You may begin when the clock reaches the 3-minute mark.
Sure. Hello, everyone. I'm Kishore from North Dakota State University, and my research is on gut microbiome. We are taught in our childhood that all microbes are bad. They cause some illness to us. But it's not so true, because there are many friendly microbes which protect us. And we acquire the right combination of these microbes right at birth. They reside in the gut and grow in colonies. We have colonies of virus, fungi, predominantly bacteria, living in the gut and collectively called as gut microbiome. These trillions of bacteria are always at war, trying to conquer gut as their habitat. In young individual, army of good bacteria outnumbers the army of bad bacteria, by which it maintains a protective environment in the gut. It acts as a barrier for foreign substances. As there are no intruders, bone marrow will not initiate an inflammation, and all the vital organs like brain, heart, and capillaries will function healthily. However, with the aging, bad bacteria now outnumbers the good bacteria. Now, these bad pathogenic bacteria secrete small particles called as metabolites. These metabolites attack the gut wall and create leaky pores in gut wall, which we call leaky gut. Through this leaky gut, small particles enter into the circulation and trigger bone marrow to release more number of inflammatory cells. Now it's like a sea of inflammation in which all vital organs are embedded, resulting in deadly diseases like Alzheimer's, atherosclerosis, heart attack, stroke, and causing mortalities. Sad part is that there is no approved therapy to treat aging microbiome. So, my research is to find the lead molecule which can reverse the aging microbiome. I have carried out experiments and I found that, yes, there is abnormality in gut microbiome of aging mice. Then I've treated the aging mice with the small molecule called as angiotensin 1-7. And guess what? This therapy has reversed the aging microbiome to healthy microbiome. This therapy supported the good bacteria to generate detoxifying agents, which has sealed these leaky pores, which has also instructed the bone marrow to reduce the number of inflammatory cells, by which it has pacified the sea of inflammation and all the vital organs resume their healthy functions, resulting in healthy aging. This result suggests that angiotensin 1-7 is a promising therapy to treat aging microbiome. And if therapies like this get approved, we are one step closer to the eternal youth. But till then, listen to your gut. Thank you so much.
Thank you, Kishore. Judges, please submit your scores. Kishore, this again, very interesting.
Yeah.
Tell us a little bit about how you got interested in this field of research and how you ended up in pharmaceutical sciences.
Oh, so pharmaceutical sciences, it was already a very interesting topic to work on. But as of now, I'm working on pharmacology, which is a little bit different than what I have done my master's on it. So pharmacology is studying the mechanisms and all those stuff, which is way more interesting than the pharmaceuticals. Mm-hmm. So studying from the basics, from the molecules, from what's happening inside. So that's how that's kind of interesting. Yeah. I can explain the mechanisms, how what leaky guts are happening, but it's 3 minutes.
No. That sounds great. Thank you very much.
Thank you so much.
Are we ready for our next presenter? We may need to, oh, here we go. Thank you. So our next presenter is Robbie Fadil. He is from the Biomedical Engineering graduate program at the University of North Dakota. His advisor is Koyar Tavakolian, And the presentation of the title of his presentation is Preventing Falls in Parkinson's Disease. Robbie, are you ready?
Yes.
You may proceed when the clock reaches 3-minute mark. Faye, could you please advance the slide and begin? Robbie, I apologize. We may be experiencing a bit of technical difficulties here. Here we go.
We in North Dakota have a lot to be proud of. We are #1 in the country for honey production, and we are #2 in the nation for oil production. But unfortunately, we are the 3rd in the country for incidence of Parkinson's disease. Parkinson's disease is a brain disorder that affects body movements, and symptoms include tremors, muscle weakness, slow movement, and lack of balance and coordination, all of which contribute to falls in Parkinson's disease. The consequences of fall in Parkinson's disease are significant and far-reaching, often resulting in injury and fear of falling when performing day-to-day activities, as well as financial hardship. The risk of falling in people with Parkinson's disease is 3 times higher than in people without the disease. To prevent falls, doctors will measure the way Parkinson's patients stand, walk, and maintain their balance, so specific weaknesses could be addressed with specific exercises and medication. However, the current equipment for measuring how patients stand, walk, and maintain their balance are large and expensive, making it challenging for doctors to have it at their clinics and nearly impossible for patients to have it at home. In my research, I am focused on developing a better way to predict and prevent falls in Parkinson's disease. The solution is to use a portable, inexpensive bedroom scale. Well, not just any bedroom scale, but an advanced one. The patient will stand on the scale and it will measure their heart rhythm, respiration, the strength of their leg muscles, and their balance. Then it will tell them if the risk of falling today is high or low. The scale will also alert the patient's caregiver or call 911 in case of an emergency. For over a year, I have been going to Sanford Clinic to record data from Parkinson's patients. From the 45 patients I recorded data from, I found that preventing falls not only depends on the way Parkinson's patients stand, walk, and maintain their balance, but also heart rate, respiration, leg muscle activity, brain-heart communication, and so many other factors. I believe very soon this technology will alleviate some stress on caregivers by assisting doctors in fall prevention. This technology will also provide a sense of independence for people with Parkinson's disease, especially the ones in remote areas who don't have access to a convenient healthcare. With further research, our device could also be used to prevent falls in healthy elderly people and those who might suffer from certain neurological disorders. Thank you for your time. It was a great pleasure to be here today. Thank you.
Thank you, Robbie. Judges, please submit your scores. And I do want to apologize again. We had a sudden internet drop, which made the slides be kind of static and not moving on our end, at least what we were seeing. So Ravi, thank you for this presentation, and tell us how you got interested in this topic to study it nonetheless.
I was always interested to working on with patients and on preventing and help people with certain diseases. So I was working in Morocco, so it's in North Africa, and I was working on breast cancer. And my supervisor approached me and told me, "Hey, Ravi, there is an opportunity that you can go to the US and work there." So I applied for that scholarship. It's called Fulbright Scholarship. And then I got it, and then I came to the US and started working on Parkinson's disease. And I enjoy really working with patients and trying to help them as much as I can.
That's wonderful. Thank you very much. And let's see if we're ready for our next presenter. Our next presenter is Dalton Miller. He is from the chemistry graduate program at Boise State University. His mentor is Ken Cornell, and the title of his presentation is Inactivation of Biofilms in Chronic Wounds Using Cold Atmospheric Pressure Plasma. Dalton, are you ready?
I believe I am.
Okay, you may begin when the clock reaches the 3-minute.
Sounds good. Thank you. Chronic wounds, seen here on your left, are wounds that fail to heal for months to years from their initial onset. They affect more than 6.5 million Americans every single year, and their treatment costs northwards of $50 billion annually. Now, obviously, that's very expensive, right? That's a lot of money. But what's significantly more costly than that is the amount of pain and suffering that people with these wounds have to go through. You see, once a week, these patients go into a wound care facility where a doctor will unwrap their wound, snag a bottle of antiseptic, and vigorously scrub out the wound site. And this is necessary to kill any bacteria or fungi present that could cause an infection. But, well, as you might imagine, vigorously scrubbing an open wound is an excruciating procedure, to say the least. Now, after this, the doctor will trim any dead tissue from the wound site, rinse the wound out, wrap it back up, give the patient a pat on the back and say, well, we'll see you in a week. Hopefully that will heal. Of course, the doctor can't actually say when or even if the wound will heal. I don't think that any of you here today would describe that procedure as ideal, effective, or even particularly good. And it's to that end that we're working on a new method for treating chronic wounds. It uses an ionized gas, also called a plasma, which you can see there on your right, and it works by generating a wide variety of highly reactive molecules that serve two basic functions in the wound site. The first of those functions is in sterilization. These molecules have the capacity to kill bacteria and fungi present in the wound site, but because we are using a gas rather than the more traditional liquid antiseptic, no vigorous, painful scrubbing is necessary. The second of those two functions is in wound healing signaling. As an example here, if you get a cut on the back of your hand, your body will begin producing these exact same molecules to tell the cells around the cut to begin dividing and thus healing the cut. By directly introducing these molecules to the wound site, we can speed up the rate of wound healing simply by telling the patient's own body to heal the wound faster. Now, right now, we're working in pig wounds to examine safety and efficacy, but our initial results are very promising. In as little as a 10-minute treatment time, which is less than it often takes to scrub these wounds out, we're seeing good sterilization of the bacteria, as well as no damage to the surrounding pig tissue. Now, we hope that eventually this will be used clinically, both to decrease the amount of pain associated with having a chronic wound, as well as, more importantly, to speed up the wound healing process so that we can give these patients their lives back. Thank you.
Thank you very much, Dalton. Judges, please submit your scores. And again, I will ask, how did you get interested in this line of research?
Yeah, sure. It was sort of a good intersection of two different things that I've always had in mind for research. I've always found medicine to be particularly compelling, and that's an area in which I've wanted to work for a long time. And in terms of research, the things that I've always found the most interesting have been the things with an immediate direct application, working on something that I would do it, and then it would be pretty immediately used in improving patient outcomes or improving quality of life. That was the area that I always found the most compelling. And this seemed to fit that best at the institution that I'm in here. And I found it to be very interesting. So
that's wonderful. Thank you very much. I wish you success with continuing in this line of research, and hopefully it will come to humans soon.
Well, thank you. I appreciate it.
Thank you. And let's see, are we ready for our next contestant? Thank you. Our next presenter is Amelia Munson. She's from the Animal Behavior graduate program at University of California, Davis. Her advisors are Drs. Andy Shee and Dr. Sean Killen from Glasgow. And the presentation title is One Big Problem, One Tiny Solution. Amelia, are you ready?
I am, but the presentation title is wrong. Would you like the correct one?
Oh, absolutely. Please do share.
The correct one is Fact or Fiction. What doesn't kill you makes you stronger.
Okay. Let's see. Apologies. Are we on the right slide? Is this your slide? Okay, great. You may begin when the clock reaches 3-minute mark.
All of my favorite stories about me from childhood involve some amount of danger. They're the times I fell out of a treehouse and then got right back up again. And I think I like these stories so much because these are the moments that made me into the resilient adult that I am today. And I think this idea that hardships make us stronger is a really common narrative. But interestingly, it's not the logic we use when we are tasked with taking care of some of nature's most valuable babies, endangered wild animals. Take salmon, for instance. When we raise this culturally and economically important fish species in captivity, we bring them into hatcheries that are really sheltered, and then we release them to the wild where they have to deal with real predators that really want to kill them. And importantly, survival is lower for captive-born individuals compared to wild fish. So the question that really motivated my dissertation was whether there were experiences that we could give fish during development that would make them more resilient as adults. So to study this, I brought Western mosquito fish into the lab, bred them, and then exposed their offspring to different challenges. So you can see here that I have this red group that was exposed to changing temperatures, this blue group that was exposed to the very scary scent of largemouth bass, the purple group that got both challenges, and then the gold group that didn't have to deal with anything stressful. And some of the effects on adults are apparent just by looking at them. You can see my middle two groups are smaller as adults. And that's interesting because these are the fish that were both exposed to predators, and smaller fish are just less appetizing to predators. But I actually cared less about what my fish looked like and more about who they were. Essentially, I wanted to give them a BuzzFeed-style personality quiz, but with questions that a fish could answer. So to do this, I measured their behavior in a variety of different contexts. And I think the most interesting thing that I found is actually also the most basic. When you take the fish and you put them in new tanks and measure their activity, the fish from the top three groups, the fish who've been exposed to just any type of challenge during development, were more behaviorally consistent. And this is fascinating because consistency is just a fundamental aspect of behavior in wild fish populations. It helps them coordinate group behavior and makes them more effective at avoiding predators. What I think is happening is that the golden group, that sheltered group, is growing up to behave more erratically, which puts them at an increased risk of being eaten by predators. I'm now curious whether these relatively simple developmental mechanisms can be used to increase real-world survival of fish like those salmon I talked about at the beginning. Because my fish are a great reminder that what doesn't kill you doesn't really make you stronger. It just makes you normal. Thank you so much.
Thank you, Amelia. Judges, please submit your scores. I am interested to know how you got into this line of research. What drew you to fish?
I think some of it is that like fish are just far more fascinating than most people give them credit for. They are just like crazy cool living this world underwater. But I think a lot of it is also that like I'm surrounded by people who have all had challenges in their life. And that's sort of a general characteristic in humans. And I was sort of curious, what does this mean? And fish were a great way to study that question.
That's wonderful. I wonder if you would have the same effect in other animals, but this is great research, and best wishes for continued success and finishing your degree. Okay, are we ready? I apologize. I forgot to unmute myself. Melissa, the next presenter is Melissa Schofield. She's from the microbiology graduate program. Her university is University of Nevada, Las Vegas. Her mentor is Bushan Teng, and the presentation title is Investigating Mutations of a Deadly Respiratory Pathogen. Melissa, are you ready?
I am.
Okay. You may begin when the clock reaches the 3-minute mark.
Awesome. Thank you. Today, I'm going to tell you about a pathogen with an interesting mutation, but I first need to talk about cystic fibrosis, more commonly called CF. CF is a genetic disease that causes a buildup of mucus in the lungs and other organs. And as shown at number 1, CF patients experience repeated lung infections from a variety of different bacteria. Now, one such bacteria is of great interest in our lab called Pseudomonas aeruginosa. This bacteria forms chronic infections in the lungs of cystic fibrosis patients. And in that CF lung environment, these bacteria acquire a mutation that causes them to become thick and slimy, as shown in the photo at number 2. We call these slimy bacteria mucoid. Now imagine trying to breathe with mucoid, slimy bacteria inside your lungs. It's not hard to imagine how this conversion from normal to slimy or non-mucoid to mucoid results in increased mortality, and this is exemplified by the graph at number 3. This graph shows lung function decline in CF patients, where the y-axis is lung function and the x-axis is patient age. The dashed line through the center of the graph shows the point at which these patients will need a lung transplant to survive. You can see patients with non-mucoid infections stay well above that line, but patients with mucoid infections will drop below that line as they age, demonstrating how deadly this conversion can be. Now, to make things even more interesting, in that CF lung environment, those mucoid bacteria sometimes acquire a second mutation, thus reverting back to a non-mucoid type. And this is shown in the picture at number 4. Now, this is thought to occur because that mixed bacterial population may be stronger than either type on its own. However, these non-mucoid bacteria are more susceptible to antibiotics because they've lost the ability to produce that slime to protect themselves. So, if we can influence this conversion, we can make a more susceptible population and better treat these infections. The problem is we don't know what factors are involved in that reversion, which brings me to my current project at number 5, where I'll be investigating different factors that affect reversion rate. I'm looking at both host factors and treatments, some of which CF patients undergo from a very young age, to see if we can find a factor that increases this mucoid to non-mucoid reversion rate. If we can increase this rate and have this happen in a larger number of bacteria, we can then better treat these infections. Better treatment of infections leads to increased survival and better outcomes for CF patients. Thank you.
Thank you very much, Melissa. This was interesting and very promising research. How did you get interested in this line of research? What was your entry point into the field?
Sure. So really it was kind of like started the first time I took a microbiology class and I was like mind blown. Oh my gosh, this affects literally every part of life everywhere. And so I became really, really interested in microbiology. And I found my PI's lab who she works in, infection models, but there's also this organism that grows in water and plants. And just there's so many different applications to studying a microbe that grows biofilms and things like that. So yeah, just roped me in.
That's wonderful. Thank you for sharing that. And of course, best wishes for success in completing your degree.
Thank you.
Are we ready for our final presenter? Our final presenter is Suk Moon, but we go by Alice Wong. She's from the Computational and Data Sciences graduate program at Chapman University. Her mentor is Yuri Maus, and the presentation title is Whodunit? Explorations of the Sense of Agency with Neurotranslation. Sorry. Let's see. Alice, are you ready?
Yes.
You may begin when the
Hello, everyone. Have you ever felt like a puppet moving without a will? And no, I don't mean existentially. I mean something more concrete, like a muscle spasm, or the weird kind of jerk you get when you're just about to fall asleep. Where does that movement come from? It's certainly your own hand or leg jerking, but it doesn't feel like you doing it. That's what it feels like when you don't have a sense of agency over your own movements. When you do, that's just normal life. My research looks at how our brains construct the sense of agency, or more formally, the subjective sense of moving and controlling your body to make things happen. But first, why should anyone care to know how brains construct a sense of agency? For one, not everyone has a typically functioning sense of agency. People with schizophrenia, Tourette syndrome, or dissociative identity disorder, formerly known as multiple personality disorder, for example, have a very inconsistent sense of agency. If we know how the sense of agency is supposed to work in typical brains, we might one day be able to alleviate the distress that comes from not having a consistent sense of agency. So, how exactly do I study the sense of agency? It's usually tricky to decouple movement from a sense of agency. After all, muscle spasms don't happen very often. But in the lab, I can do this with electricity. Using electricity, I can stimulate the portion of your brain that controls your thumb and make it twitch involuntarily. This feels like the knee-jerk reflex you have when a doctor taps you just below your knee, except in this case, the stimulator taps your brain and the reflex makes your thumb twitch. Normally, this is no big deal. I stimulate your brain, your thumb twitches. Fun, but not too interesting. You weren't intending to move your thumb anyways, so you have no reason to think that the thumb movement is coming from you. But what if I ask you to voluntarily twitch your thumb at the same time, and I don't tell you when the stimulation is coming? Do you think you could tell who or what moved your thumb? Spoiler alert, my results suggest that you can't always tell, especially if the stimulation comes around the same time as your own intents to move. What could these results mean? Well, if there's a predictable time window where stimulation-induced movement and your own voluntary movement can be confused with each other, it may be possible to make voluntary movements feel involuntary, or vice versa, make involuntary movements feel voluntary. Further brain imaging research could then help us understand the difference in how the healthy brain represents voluntary and involuntary movements. In the end, this research could be the foundation upon which neurological and artificial prostheses can be developed to restore a proper sense of agency in those with a disordered sense of agency. My name is Alice Long, and this is my 3-minute thesis.
Wonderful, Alice. Thank you very much. Judges, please submit your scores and tally the final results. Alice, how did you get into this line of research? It is fascinating.
Yes, my PI has a program of research that looks at voluntary motion, action, and free will in general. And me personally, I've been more interested in the cognitive side of the human cognition and the brain sciences. So when you couple that with voluntary action, what you get is kind of investigating the perception of voluntary action, which cashes out into sense of agency. So yeah, this project, I just jumped at the chance when I had the chance to do so.
That's wonderful. Thank you again. This is amazing research. I'm always fascinated by neuroscience, so very interesting to me. I'd like to invite all of our presenters to unmute themselves and appear on screen. We can give the judges a bit of time to tally their scores. And in this time, what I would like to do is ask you whether this preparation for the 3-minute thesis presentation gave you a chance to better understand your research or better refine your research. Any thoughts you may share?
Yes, definitely, because we read a lot to write this the script for the 3-minute thesis. In a course of reading, we read the reviews, research, and everything. So it has helped me to understand what are the experiments to be done for the future studies.
Thank you. Yes, Alice.
Yeah, the most interesting thing in this process of doing the 3-minute thesis to me that was super helpful was kind of clarifying my thought, because in order to compress the amount of information you have into a space of 3 minutes, you have to really be able to tell what's important and what's not. So, and I found that talking to individuals who weren't in the field or anything like that, they were like my friends from college in a different field entirely. That was actually the most helpful thing in trying to calibrate the amount of information that goes into the thesis. There's some amount of planning and thinking you can do, but until you get feedback, I was very surprised by the feedback, for example.
Wonderful. And Amelia?
Yeah, I totally agree. I feel like it was the same experience for me. I definitely spent a lot of time, like every time I was in an Uber, being like, ooh, let me try out a different pitch on this person and see how it goes.
Great idea.
That's wonderful. I just got news that we have our judges ready with their final tally of the scores. And let's see. And we have, Pei, can we share our final screen? I would like to congratulate our winners. For first place, we have Dalton Miller. Second place, Alice Wong. And third place is Amelia Munson. You are all excellent. I want to congratulate the winners with my most sincere congratulations, but having all of you here is such a joy, and I really, really appreciate your excellence and your research and your enthusiasm for your research. So thank you so much for being here. Thank you for your presentations. I'd like to thank the judges for their work and our technical support. I'd like to thank the audience for joining us. And I'd like to remind everyone to join us for our final discussion groups, which start after just a short break here, 15 minutes on the agenda. Please do log in for our final discussion group session. Once again, thank you all, and I wish you a very successful conclusion of your studies.
Thank you.
Bye.